And this is the chest X-ray of the bilateral alveolar infiltration in the lower zone. And the immunofluorescence here of the IgG type against the glomerular basement membrane and adjacent tubular basement membrane. Another disease is myasthenia gravis, which is also a type 2 hypersensitivity reaction, which is a result when the body form autoantibodies against the acetylcholine receptor on the skeletal muscle, which is lead to relaxation. Here, most significant muscle relaxation is the muscle of the upper eyelid, which is lead to ptosis, which is either bilateral or unilateral. Another disease, autoimmune disease, Hashimoto's thyroiditis, which is a chronic lymphocytic infiltration of the thyroid gland, and the immune system will attack the thyroid gland, result in inflammation, and lead to underactive thyroid gland, which is hypothyroidism. It is mixed of type 2 and type 4 hypersensitivity reaction, which is caused by the T helper 1 and T-delayed type hypersensitivity and autoantibodies against the thyroid protein and the thyroid cells. And here we found that the infiltration of the lymphocytes, histopathology infiltration of the thyroid cells by the lymphocyte. So during the histopathological examination, we have a gritty sensation when we cut the thyroid gland by the knife. And here the immunofluorescence against the thyroglobulin and the thyroid cells. The another autoimmune disease, which is the insulin-dependent diabetes mellitus, which is type 2, type 4 hypersensitivity reaction, which is caused by the T cytotoxic cell that infiltrate the pancreatic cell and the T-delayed type hypersensitivity cells, and also formation of the autoantibodies against the pancreatic beta cell. And here the immunofluorescence study of the autoantibodies against the pancreatic beta cells, which give fluorescent apple green light. And another disease of the autoimmune disease, which is the non-organ specific autoimmune disease, which includes the first and most important is systemic lupus erythematosus, which is SLE, which is the abbreviation of systemic L for the lupus and the T for the erythematosus, which is type 3 hypersensitivity reaction, which is non-organ specific caused by the autoantibodies against double-strand DNA. This is very important. No single strand is against the double-strand DNA, and autoantibodies against nuclear protein, RBCs, platelets, membrane, and the immune complex are deposited in the kidney. And here we can it's called systemic because it's affecting multiple organs: the lung, the heart, the kidney, the joints, and also the skin. We found butterfly rash on the skin. The another disease, which is rheumatoid arthritis, which is type 3 hypersensitivity reaction, which is also non-organ specific autoimmune disease. The cause of this disease, the autoantibodies, which is the IgM against the IgG, and autoantibodies leads to immune complex formation that deposit in the synovium of the joints. The genetic factors also contribute in this disease, which is HLA-DR4 and DR1. And here the joints, when we magnify this or make zoom to this area, we can find that the this is the blood vessels and migration of the cells and secrete T helper cells, T helper 17 cells that secrete the interleukin 17, which is lead to the synovial. This act on the synovial fibroblast, and also the stimulate the synovial macrophage, and the synovial T cells, T helper 17 cells will be aggregate, and this lead to secretion of inflammatory cytokines like tumor necrosis factor, interleukin 1, interleukin 6, and this lead to increased rankle, and this lead to stimulation of osteoclasts that destroy the joint and lead to increase the activity of the osteoclast cell that lead to osteoclastogenesis and bone destruction. So we found that the joint will form a soup of the cytokines, and it affects the small joints, interphalangeal joints. And this is the explanation of previous figure. And the autoimmune response in rheumatoid arthritis, the autoantigen is the IgG, which is called the rheumatoid factor, type 2 collagen of the cartilage, and the newest one, the cyclophosphamide protein, is CCP, which is highly specific for the rheumatoid arthritis, and when it is positive, indicate bad prognosis and aggressive and persistent disease. Treatment by the non-specific suppression of the immune system using the corticosteroid in high dose and the chemotherapy, azathioprine, and cyclophosphamide. And we must concentrate on the immunotherapy that only suppress the immune system and not all other highly dividing cells. So we use monoclonal antibodies specific to CD4 T cell receptor, MHC, interleukin 2 tumor cross factor. And as you know previously, this is the diagram show us the stimulation and activation of the T cells, and this is the antigen presenting cells and T cells, and this is the receptors which is involved in the stimulation of the T cells, and we found autoantibodies against these receptors. And peptide blockade of the MHC molecules by synthesis the peptide, which is located in this space, instead of the cell antigen. An induction of tolerance by the oral antigen, and which is cheap and easy to administer for application in human disease. And rheumatoid arthritis, one of the autoimmune diseases that had benefit from this form of immunotherapy using mainly collagen type 2. Another transfer of Treg cells can inhibit disease pathogenesis. Another method of treatment, blocking antibody against interleukin 17 and interleukin 23. Statin that reduce cholesterol, found that decrease cytokine level, pro-inflammatory cytokine, and decrease adhesion molecules expression. The last one is generating immune tolerance by using tolerizing DNA vaccine using molecular methods. DNA plasmid encoding cell antigen is translated into dendritic cells, and the presentation of cell antigen by antigen-presented cells without adequate co-stimulation leads to energy or tolerance of T cells because of the lack of interaction between the CD28 with CD80 or CD86, which is the second signal of the T lymphocyte activation. And here, figure show us this is the plasmid and encoding the cell antigen, and this is the dendritic cell, and this is the cell. This dendritic cells will express the cell antigen, and there is no stimulation to the second signal CD28, so there is tolerance. While here the immune activation, this is the non-cell antigen, and this is the dendritic cells which express the non-cell antigen, and this is the first signal of the T lymphocyte activation, and this is the second signal of the T lymphocyte activation, and this is lead to activation of the T cells, which is the co-stimulation. While here in the tolerance, there is no activation of the second signals, so end with tolerance. And thank you.